So here we are again, looking at that. Right? ATP is bound, right? Hydrolysis puts it in the cock position, and then there's binding, right? But I'm sorry, C what happened, the interaction happens, and then the power stroke. But here's where we get into the second part, right? That's ATP. That's the role of ATP in this. Now we want to look at the role of calcium in all this. And what we're going to see is that this interaction between the myosin head and the actin is calcium dependent. Right? So here's the whole cycle again from a different book, right? And, myosin head hydrolyzes ATP, it's cocked. And at this point, once the head is cocked, it's right close to the actin. Myosin might be binding weakly to actin. We don't have to worry about this detail. What we're concerned here is the calcium signal over here. That's going to allow the strong interaction and then that consequent pivot over there, right? This calcium signal. So what do we want to know about this is where is this calcium signal coming from, right? When and how is calcium present and what does it do that allows this interaction? We will see that on the actin filament, there are particular myosin binding sites, right? Which are only going to be exposed in the presence of calcium. Okay, they're only going to be exposed, these myosin binding sites. If calcium isn't there, the myosin head cannot find those myosin binding sites. Again, demonstration right here, right over here. My watch is the myosin binding site. It's hidden for some reason, right? Calcium comes along and it's all of a sudden it's exposed and then we can do that pivot thing. So that's what's going to happen here. So let's look a little bit closer here. Why is cross bridge formation dependent on the calcium? Right, so here we're going to look ahead at that myosin head, right? We already saw that ATP binding site, right? It had that ATPase activity. But here we have another part of the myosin head that is an actin binding site. Right? And that's right over here. And again, on the actin filaments over here, right? These little balls, you have like the actin is formed by these string of actin, the G actin, right, which forms this F actin over here, right, like two strings of pearl, right? But on these little balls right over here on the actin, you have the myosin binding sites on actin over here, right? So you have an actin myosin binding site on the myosin binding site on the actin. Makes sense, right? They need an interaction, right? And here's that hydrolyzed ATP. It's in the cock position. So here are the regulatory elements that are also wound up with the actin filament. First one here, the string that's going along the length of the actin filament is tropomyosin. Tropomyosin, right? One of the regulatory elements. What the tropomyosin is doing is keeping these myosin binding sites, right, which you can see are in a particular sort of orientation. They're going to keep them away from your actin binding sites of the myosin. That is, they are what's going to kind of keep them in a position away from the myosin heads over there, right? So tropomyosin is kind of stopping that interaction just by the way it kind of twists the actin filaments around. And then there's a second regulatory element in blue right over here called troponin, right, or troponin. These ones over here are going to be what interacts with the calcium. When it does, what's going to happen? Calcium is going to come in, bind to troponin. Once that interaction happens, troponin is actually going to move the tropomyosin away so that the myosin head can actually find those myosin binding sites. Right? So in the presence of calcium, the interaction of troponin and calcium is going to move the tropomyosin away from the myosin binding site, right? So we have calcium binds to troponin. Troponin then moves tropomyosin away from the myosin binding sites on actin, right? And so The next question you want to know, this is why calcium is important. The next question you want to know is where is calcium coming from right here? And you'll see that right before the contraction, calcium is going to be released from the sarcoplasmic reticulum that is going to be surrounding the whole sarcomere. We'll put it all together at the end, right? We're just going into detail over here. So now we go back to the sort of microanatomy, right? The anatomy within the myofibril right here. Again, packed with the myofibrils, but they also have all the organelles, right? And those specialized organelles, right? Remember, cells have the endoplasmic reticulum. Muscle cells have the sarcoplasmic reticulum in blue here, right? So what you see is the sarcomere over here. Then you see kind of bookending the sarcomere here are these light blue things called the T-tubules over here. And then on either end of the T-tubule over here is the portion of the sarcoplasmic reticulum called the terminal cisterns, which are going to interact with these T-tubules. Okay. So your sarcoplasmic reticulum is where the calcium is stored, right? And the sarcoplasmic reticulum, along with these T-tubules here, is kind of covering the whole sarcomere, right? So you could imagine all the sarcoplasmic reticulum has to do is dump all this calcium on here, and that will cause that cross bridge formation. Makes sense so far, right? The storage and this anatomy of it from afar. Here's a more cartoon version, right, with some detail over here. Here's the T-tubules. What are the T-tubules? They're invaginations of the sarcolemma. That is, here's the membrane of the muscle fiber, and some of the part of the membrane dips deep down into the muscle cell, right? Deep down, but it's a continuation, right? And here's the membrane continuing on. It goes deep down to the muscle cell, continues on here, and then dips down into here, right? These are the T-tubules. So whatever's happening in your sarcolemma, your plasma membrane, is going to happen in the T-tubules. Okay, that's an important point. And then the T-tubules are going to be in contact, again, on either side of them. They're going to have these terminal cisterns, right? So your T-tubule and your flanking terminal cisterna make the triad. Three things, right? T-tubule and the left and right terminal cistern. Okay, sarcoplasmic reticulum, what does it do? All sorts of calcium, right? Just waiting to be poured onto here so that these can interact. Everybody with me so far about this structure? You're waiting to see how does it happen. Okay, so just to go back right here, here is that actin filament with the tropomyosin and the troponin complex, right? And some conformation. And then the calcium is what's going to be needed to twist it around so that the myosin head can interact. Okay, so we're just waiting for this calcium. We know where it's coming from. How's that going to happen? Well, there is going to be in this arrows right here. You see these arrows going down here. There is going to be a magical signal traveling down the surface of the entire muscle fiber that's going to go down into this T-tubules. And that signal is going to be what causes the release of calcium, right, on spill onto this complex right there. With me so far? Now we want to know, what is this magical signal that causes this release, and how does it do it? This magical signal is what's known as an action potential. An action potential traveling down your cell membrane, right, your sarcolemma, and then down the T-tubules. Right, so there's going to be this electrical impulse traveling down here and here, which is going to ultimately cause this release. So now we're going to break that down further. How does that happen?